First of all, we're very, very excited about this program. We ran the Summer Institute also last year, and we thought it went great, but we think we've improved it a lot from what we've learned from last year, so we think it's going to be even better this year. And we'd love to hear from you, not just at the end, but throughout the 2 weeks, about what you think, what we can do to improve. We want to make it the best experience we can for you, so please give us feedback. The more the better, positive as well as negative. We're grateful to lots of different people and institutions. So the Russell Sage Foundation is funding the workshop. They paid for your stay here, your flights, your food, and so we're all very, very grateful to them. Liana Chatrath from the Russell Sage Foundation helped to organize this event. She's not here, but we're nonetheless grateful. Samantha Cherney, who is here and who all of you have met. Raise your hand, Samantha. So Samantha is my wife, and she's also the logistics director for the SSGAC, and she, as you all know, is doing a tremendous work for all of us. AdHealth is the data that we're giving you access to. It's not a normal thing for a data provider that has such sensitive data as genetic data is to make it available to many students for just a 2-week period. So we're extremely grateful for them for doing that. The National Bureau of Economic Research is giving all of you access to the server that you're using to access the AdHealth data. That's also highly unusual because there's lots of privacy and IRB concerns, but they were willing to help make that work. And we're grateful to all of the instructors, all of whom are volunteering their time to be here because they're as excited as I am about this program and making sure that you get a lot out of it. Okay, so what is social science genomics? Well, it's just the use of genomic data in social science research. And you'll see lots of examples of that. We think that there's lots of reasons to be excited about that, and that's what today's lectures are going to be. But there's also a lot of challenges to appropriately using the data, interpreting the results that come out of it, and communicating those results correctly and effectively. And tonight's lecture, which will be during dinner, is going to be about the ethical issues in using, interpreting, and communicating results. And we think that's always important in research, but it's especially important for research that involves genetics, and especially for genetics research that mixes with social science, because there's a pretty bad history from the past of the combination of those two fields. Another aspect of ethics is research ethics. And so a lot of the program is also going to be about doing research well, which involves doing it ethically, responsibly, being transparent about what you've done, and making sure that you're doing results that are scientifically appropriate. Okay, so why have a whole course about genetic data? Well, there's a number of things that are unique, or at least distinctive, about genetic data. So one thing is that different DNA is fundamentally what makes us different people from each other. So if we're interested in heterogeneity across people, this is kind of an important starting point. Genetic data is unlike much other data about people in that it's predetermined. At conception, when the sperm and the egg come together, your DNA is determined. It's not going to change for the rest of your life. And that is pretty useful for a lot of for a lot of purposes. You don't need to worry about endogenous effects of other things that happen changing your DNA. It may change gene expression, and that's actually quite important, and we'll be talking about that. But the DNA itself is not going to change. Another really important, interesting thing about genetic data is that it's randomly assigned at conception, conditional on who your parents are. So it's effectively like a natural experiment, what DNA you end up getting. And that gives us leverage to address causal questions that are often very hard to answer in the social sciences, where you usually don't have randomization. Another interesting thing about genetic data is that the universe that we need to explore is finite and, at least in principle, fully measurable. You can sequence the DNA, and then you know all of the DNA that a person has. It's not like a lot of other things we study, environmental variables, where you might have some measure of it, but that's just measuring an aspect of it, and it's missing some things. In principle, this is data where we know exactly the whole picture. Another interesting thing is that the biological function of the DNA is knowable. Now, often we don't know it. In fact, our knowledge about the biology of DNA is about what the DNA does is fairly primitive at this point in a lot of cases. But in principle, there's this other science we can go to, biology, to learn about mechanisms. And we don't just need to rely on kind of social science methods of trying to tease out mediation. It's also, so genetic data is an example of big data, which itself is important for various reasons and is very popular these days. And genetic data comes up all the time in discussions of big data because it's a leading example. So a lot of what you're going to learn about genetic data carries over to other contexts of big data. And genetic data, this is sort of related to some of the other points, genetic data has some distinctive features that are well characterized. So we'll talk about a number of them. It's things like it's a 0, 1, 2 variable. There's correlation structure in the genome, which is well characterized. And so part of the reason why you need 2 weeks to learn about this kind of data is because there's a lot of those details to understand and then learn how they relate to applications of the data. OK, so why are we having this course now? Why didn't we start this Summer Institute 5 years ago or 10 years ago? Well, we couldn't really. Now, in the last few years, there have been major advances in the measurement of genetic variation. In particular, it's become much cheaper than in the past. So here is a figure that NIH puts out that shows the cost per megabase of measuring DNA. And so notice the y-axis here is on a log scale. So a linear decrease on that scale is actually an exponential fall in the cost. What's graphed here in this line is Moore's law. That's the rate at which computation speed has increased. And that's often given as an example of really fast falling cost. The dots here are the cost of the genetic data, and you can see on average the increase has been faster than Moore's law. In fact, the cost has been falling roughly by a factor of 2 every year. So now, we'll talk more about these kinds of data, but the genome-wide data that we use in a lot of work in social science genomics costs about $40 to $50 a person to measure. Last year it was $100. Next year it's going to be, more like probably $25 or $20 if we were to guess. Okay. So as a result of that falling cost, the genomic data are now increasingly available in data sets that social scientists use regularly. So like the ad health data that you have access to for this course, it's also in the health and retirement study data. Dalton is leading efforts to get it included in the PSID, the panel study of income dynamics. And many other data sets that we'll be talking about. It's also possible to measure it yourself, potentially, if you want to go collect your own data, run a laboratory experiment. You can think about collecting genetic data pretty easily now. There have also been, in the last few years, dramatic advances in our understanding of which genetic variants are associated with which behavioral traits, or how we can predict behavioral traits based on genetic data. And so that means that this data are now much more useful than they ever were in the past for doing social science research. We're also now at a place where the discoveries are mounting very rapidly. And in fact, our ability to predict behavioral traits is increasing at an extremely fast pace. We seem to be at a cusp in terms of what kind of sample sizes are available that are allowing us to go from the realm of predicting, for example, educational attainment at 1 to 2% of the variance as of a couple of years ago to, in the next year, closer to 15% of the variance, 10, 11, 12% of the variance. And that now we're talking, extremely predictive variables that have potentially lots of uses. So it's the time has really come for doing good research in social science genomics that can be well powered. There's lots of new opportunities that have only now become possible. And so this is the perfect time to be learning these tools and thinking about how to incorporate it into your research. OK, so who are we, the instructors? We're a group representing a variety of different disciplines: economics, sociology, psychology, political science, genetics, bioethics. Everyone who you're going to be hearing from is among the leaders in doing research in this area. Some of us, Dalton and myself, are members of the Russell Sage Foundation Biology and Social Science Working Group, which has spawned this Summer Institute as part of the work that it's doing. And I want to mention on behalf of the Russell Sage Foundation that RSF also gives grants. In fact, there's a call for grants for doing biology and social science. And they support visiting scholars. So those of you who are in faculty positions, you could think about applying to the Russell Sage Foundation to spend a year in New York with your salary paid, no teaching. And so, go to the RSF website to find out about these opportunities. Biology and social science is one area that they are pushing now. And so, they're sympathetic to the kind of work we're going to be talking about. Many of us who are instructors are also associated with the Social Science Genetic Association Consortium, or SSGAC. The SSGAC is a group that coordinates large-scale genome-wide association studies of behavioral phenotypes. We're interested in creating tools like polygenic scores, which are indexes of genetic variants that you can use to predict traits, and make those available to researchers in different data sets. The SSGAC is also dedicated to promoting good research practices. That means things like ex-ante power calculations, pre-registration where possible, and complete reporting of your methods, and transparent reporting of all the analyses you do, publicly posting the results, and accurate communication and interpretation of findings. So you'll be hearing more about the SSGAC, and you'll see a lot of these messages kind of coming through in the 2 weeks. The organizers of this particular Summer Institute are myself and David Cesarini, who you'll be hearing from later today. And Philip Kollinger is a co-director with us of the SSGAC. Phil is at the Vrije University in Amsterdam. I'll tell you a little more about myself when the lecture on gene discovery later this week. I'll tell you about how I got into this kind of research. I'll just say for now that my own background is in behavioral economics. I got my PhD in economics in 2006. I am currently an associate professor at the University of Southern California in an interdisciplinary group called the Center for Economic and Social Research. And David and I are both alumni of the Russell Sage Foundation Summer Institute in Behavioral Economics. There's also a third summer institute that they now sponsor in addition to behavioral economics and social science, genomics, and computational social science. Okay, so who are you guys? Well, you heard last night when everyone gave introductions. You have a sense, I think, of the mix of disciplinary backgrounds. You are a selected group of the applicants. It was actually an extremely strong group of applicants, and we're really excited about all of you. We thought, you were a tremendously great group this year, and we were looking for in the application process were people who we thought had the potential to be future leaders in your own discipline. And so, you should take it as a real compliment that, we thought that's you. This is the mix of disciplines. I think we have pretty good disciplinary diversity. This includes only the actual students who are taking the course for credit. There's also a few auditors from various disciplines, and so you'll meet them at lunches and dinners, and hopefully they can, they're just as much part of the conversation as everyone else. And you should definitely make sure to talk from each other, because I think chances are pretty good you'll learn more from each other than you will from any of the instructors. Okay, so what's the point of the Summer Institute? Well, one thing, the most important thing is to help you develop your own research ideas. So that's part of why we're giving you access to real data and encouraging you to explore it. And on the last day of the Summer Institute, there's actually 2 70-minute long sessions that are devoted to discussing your research ideas. So during this 2 weeks, think about research, what you would like to do, how you think you could apply some of these ideas in your area, and come up with ideas so that we can talk about them on the last day. Another purpose of the Summer Institute is to help you learn how to implement and interpret some of the new methods that have been developed recently in social science genomics. Even if some of the methods you're going to learn about are not going to be ones you're likely to use yourself, but it's still important to learn about them so that you can be a consumer of the social science genomics research. Both to evaluate it for potential use in your research in the future, and also because you may be refereeing papers that use some of these methods, or you'll see them presented at conferences, and you're going to want to know, how to evaluate this work. You'll also learn about some useful data and software, and about good research practices. Okay, so let me say a little bit about our teaching philosophy. So first of all, modern genomics, there's a lot of theory that we do think is important to understand, but ultimately the field is empirically driven. And so, so that's why we worked hard to give you access to the real data and to the powerful hardware on the server and the software that's specialized for genetic data that you'll be learning about to do that kind of analysis. But I think something which is so one distinctive thing I think about this training program as opposed to some other training programs in genetics is the fact that you'll have access to the real data. I mean, I think that's really valuable, and it also means you can be exploring this data not just to do the problem sets, but out of your own curiosity to see what kinds of patterns or relationships might be there in the data, test your own hypotheses, and develop your own research. Another thing that I think is distinctive about this Summer Institute is that we put a lot of emphasis on theory. I mean, I think that comes through already from the problem sets that you've worked on. And we think that's actually really important because too often we see research in social science genetics, but even in genetics more broadly, that is, that has mistaken interpretations or just doesn't build on kind of a solid theoretical foundation. And if you're going to be able to interpret the results correctly or even ask the most interesting research questions, you really need to understand the principles, the fundamentals of the theory. So we've tried to focus mainly on the key theoretical issues and the simplest models that that convey those ideas. And so that's why we have the problem sets. They're really central to the program. We understand that there's some heterogeneity in mathematical background, and that the problem sets may be easier for some of you than for others. I urge you to struggle through the problem sets, even if they're hard, and also work with the other students. That's a big part of the learning process too, is having helping each other, and I think you'll get a lot out of the problem sets, even if they're a struggle. We also, to make life easier for you, we've tried hard to have uniform notation across the problem sets and across the lectures and the problem sets. We're not going to be perfect on that, but hopefully that'll help. So the Summer Institute is intended to be an immersive experience. It's certainly going to be immersive for us, the instructors. We've put on hold all of our research and other commitments for these 2 weeks, and our entire focus is making this the best program we can. For you, you'll have the lectures that'll be most of the days. You'll have the problem sets that are going to take a long time to do. And we hope you'll, in addition, be spending a lot of time thinking about research ideas and talking to us and to other students about potential ideas. So I encourage you to go to office hours. There's a number of office hours that are scheduled during the 2 weeks, but there's also just time during lunch and dinner when instructors are going to be around, and you should talk to us and make sure to ask us your questions or talk to us about potential research. I mentioned you should work in the problem sets in groups. I think that's a big part of the learning experience, and one of the valuable things about having in one place all of these potential leaders of the field in the future is that you are each other's potential collaborators. So also talk about research with each other with an eye toward potentially finding people to work with. We're expecting everyone to participate in everything. In fact, it was a condition of being admitted to the Summer Institute that everyone needs to be in attendance for the entire 2 weeks. We will provide you with every meal except for the weekend. So you're on your own starting on dinner this Friday night, and then the food here resumes again for, is it?
Sunday night dinner.
Sunday night dinner, good. If you have any logistical questions, you should email Samantha at the RSF email address that you all have. You can also, and if there's an emergency, call Samantha's cell phone number or my cell phone number. And we'll also be posting all of these slides online and give you the link so you can look up any of this information, including our numbers, later if you don't get them down now. Okay. So about the problem sets. I've already said a little bit of this already. They're going to be a mix of theory and practice. So mostly so far what you've seen is the theoretically oriented questions where you're just deriving things mathematically. As the 2 weeks go on, there'll be more computational questions that will use the ad health data, where you'll have to actually use some of the software that you'll be learning about. As I mentioned, they're supposed to be challenging. One thing I want to remind you to do is to check your answers against the solutions. So I think too many of us often make the mistake of doing a problem set and then turning it in and then never looking at it again. But there's actually a lot to learn from seeing what you got right and what you didn't, where there's some subtlety that you missed, or maybe just one question that you had skipped, and just learning what the way to approach that kind of problem. So make sure to check your answers against the solutions after we send you the solutions. For this week, as you know, there are 2 teaching assistants, both of whom are alumni of the Summer Institute from last year. They are Robbie and Megan, so raise your hands, Robbie and Megan. Okay, so that's Robbie and Megan. They'll be they're around for the full 2 weeks. If you have any questions about anything, you should feel free to talk to them. Also any other instructors, but Robbie and Megan are also among the instructors. And they'll be presenting special TA sessions that cover a number of the practical issues of accessing the server and using the software. Don't hesitate to talk to Robbie and Megan. They're really, really nice people. OK, about the computing. We're going to give you access to a bunch of different things. The cleaned data that you'll use for doing the problems. So there's lots of steps of cleaning data, and we'll talk about some of them in the lectures. You're going to have access to data that's already been cleaned. But you'll also have access to the raw data that AdHealth makes available to any researchers and the code that we use to clean the data. So that's potentially useful because you can then see what it looks like to write code that cleans the data and what the issues are that how we solve them. You'll also have access to the AdHealth codebook, which has information on all of the variables, and to the NBER server, which holds this data and the software that you're going to need to use to analyze it. You should have already gotten from Robbie your password to the server. We're only giving it out in person and on paper. Don't put it in any kind of electronic form that could be viewed by someone else or that's accessible to the internet. If you do not have does anyone not, Robbie, did you get?
There's still a fair number of people that haven't put that password yet. So just see me sometime today and we'll give it to you.
OK, so make sure to see Robbie. I want to really emphasize these 2 bolded, underlined points. Do not download anything from the server at all during this while you have access to it. And you're responsible for data security and respondent privacy. There's all sorts of legal contracts. You signed a contract saying that you would not do these things. We have a legal contract between the NBER and AdHealth saying that's not going to happen. NBER is taking responsibility for the data. We'll get in huge amounts of trouble if if any of this happens, and people running the servers can see what's going on with use of the server. So it's not something that can be done surreptitiously. Yeah.
Just to clarify that, when you say don't download anything, the typical restrictions for AdHealth are that you don't download analytic files, but that you can download things like do files or our program files that you're using to write the code.
Yeah. Robbie, do you know what the?
So nothing should be downloaded from the server at all. Something from the web server shouldn't be downloaded. If you find some potential code that might be able to help you with the data set that you're thinking about, you're of course welcome to save your own do file as long as it can't be connected to the server.
Sure, of course.
Okay. Okay, so I mentioned that there are some scheduled office hours, and also, we're always around informally. Several of us are going to be around for the full 2 weeks. So David and I and Patrick, Megan and Robbie, and also Patrick Turley. Why don't you raise your hand, Patrick? We five will be here the whole time. And there will be, various other instructors will be in and out, usually for the amount of time that they're teaching for, but some people are staying for longer. James, you're staying for the full 2 weeks?
You asked me to.
Oh, yes. I did ask you to. Yes. Fantastic. Oh, good. Then I should have mentioned James up here, too. Okay, so James Lee will also be here for the full 2 weeks. Fabulous. Dalton, you're just here for a couple days, yeah?
Tomorrow, yeah.
Okay, so Dalton is a scarce resource, only here till tomorrow. Make sure you talk to him before then. How about Raymond?
To the end of the week.
To the end of this week. Okay, so Raymond is here for this first week. And then and Fleur, you're here for this first week as well. Okay, and then there'll be other instructors coming in. So make sure to take advantage of the instructors while they're here. All right. So any questions about any of the welcome issues? Yeah.
For us who are lazy, is it okay to use SSH keys instead of having the password and having to type it in every time?
So I think we prefer that you type it in every time, just to make sure things are as secure as possible. I know it's kind of a painful password.
Sorry about that. There's real, this is real, IRB and privacy stuff that we need to make sure to follow all these rules to the letter. Okay. So, did you want to say something? Okay, no. All right. So now I want to dive into some real material. This is, I think, the core. One of the core things we want you to understand about what the basic model is underlying a genetic effect, like what we mean when we say that there is a genetic effect on something, and also what we mean by the additive model, which is going to come up a lot. So this is what I'm going to show you is essentially a generalization of what you worked out in problem set 1. I'm going to show you kind of the full version, the genomic version of what you did in problem set 1. Let's consider some outcome. So the example we're going to use a lot over these 2 weeks is educational attainment. That's going to be our outcome Y, and I indexes individuals. There's at each locus, J, there's a genotype, or genotype score, which is Xij. So that's individual I's genotype at locus J. That's going to be a 0, 1, 2 variable. You'll hear later from Fleur why genes have this, why genetic data has this particular form. But it's the number of, say, minor alleles, 0, 1, or 2. And then we have, we could consider the set of, for individual I, the set of genotypes throughout the whole genome. So this is person I's genotype at location 1, location 2, up through location capital J. We'll call that the genotype vector. And what we're almost always interested in when we talk about genetic effects are causal effects. So what do we mean by the causal effect of some genetic variant? We mean the outcome of a hypothetical experiment where at conception, so say it's locus number 2 we're interested in. At conception, we're going to go into the person's DNA and change their genotype at locus number 2. Experimentally modify it, change it from a 0 to a 1, say. And then we want to know what then happens later in that person's life at whatever, if we say it's educational attainment, maybe age 30, how much education does the person have at age 30, before and after, if we had left it at 0 or had changed it to 1. So that's what we mean by a causal effect. Now, obviously in humans, you can't do that experiment. You can actually in animals. So we're going to need ways of estimating what that causal effect is without actually doing that experiment. But it's important to understand that is the experiment you should have in mind when we're talking about causal effects. That's the thing we're trying to get at. Now, looking at the correlation between the genotype and the outcome may not give you that causal effect. So why might it be that your genotype is predictive, say, of your educational attainment, but not necessarily that's not necessarily a causal effect? Give me an example. Yeah.
Like this kind of chopsticks examples where it's sort of your genotype is predictive of some cultural group or something, and that group is much more likely to use chopsticks.
Exactly. So that's a great example. It's a famous example of if you're looking for a gene for the use of chopsticks, any gene where the genotype frequencies differ across Asian and non-Asian populations is going to show up as associated with chopsticks use. But it's obviously not causal because chopsticks is caused by being in the culture of, that uses chopsticks. So if, for example, everyone from an Asian descent has a 2 at locus number 2, and everyone of European descent has a genotype of 0 at that locus, that's going to look like that it's going to be an association, but it's not causal. OK, so how can we get at a causal effect? There's basically 2 ways. One is to study a population where, in fact, the genotype is randomly assigned. That effectively means doing this kind of hypothetical experiment, and that isn't really possible with humans. So what we end up doing to study causal effects is number 2, which is to include some control variables, which we'll call a vector z, such that once we condition on z, x is as good as randomly assigned. So these slides were supposed to be, bullet point by, reveal themselves one by one. I don't know why on David's computer they don't work that way.
PDF
Oh,
I was worried about the Mac transfer. Yeah.
OK, so I can't ask you this as kind of a quiz one by one, but I'll do my best to do that, something like that anyway.
If you want.
It's too late. This was.
Yeah.
Yeah.
This is a big moment.
Yeah. So why is the ideal thing to condition on parents' genotypes? Why would that give you as good as randomness in which X you get? Yeah.
Because once you have the parents' genotypes, it's literally randomly assigned. What you get, end up conditional on who your parents are is random.
Exactly. Once you have the parents' genotypes, it's literally random which genotype the offspring gets. Okay, so that's a perfect experiment for our purposes. It's another thing you could do is have family fixed effects. So actually, someone should explain what does that mean. Everyone may not be familiar with that term. What does it mean to have family fixed effects? Right. So you have a regression. You have family members, say a bunch of sibling pairs. That's a typical example. And you include in the regression a dummy variable, an indicator for which family that the individual is from. Okay. Why is that also an ideal experiment for our purposes? Yeah.
If you're comparing two siblings, for example, then it will also be random what their genotype is because you condition their parents' genotype.
Yes.
If they have the same parents.
Yes, exactly. So if you have siblings in your data and you control for which family they're in, then what that means is that you're looking at the differences between the siblings in their genotype, and looking at the association of that difference with the difference in their outcome. Okay, and that difference in genotype is random because they had the same parents, so they were equally likely to get different possible genotypes. And so we again have our ideal experiment where we can infer causation. What if you don't have family members, you don't know who people's parents are, and so you control for ethnicity? Why is that not going to be as good of a control? I mean, it's going to deal maybe with the chopsticks example, if you have Asians versus non-Asians. Yeah?
Because people could be genetically very diverse within an ethnic group. So
They could be diverse, but diversity in itself is not the problem. The issue is that we want that diversity to be as good as random diversity and not correlated with other factors that are influencing the outcome. Yeah?
Well, the different family environments might also confound the effect of the genetic information. So
Yeah.
within the same ethnicity group, different families might have different education philosophies of their kids. That might also affect.
So that's true. So different families could have different education philosophies that end up affecting their kids. Why does that create a confound for differences in genotypes? For inferring causation from an association between a genotype and an education of the child.
For example, can I still answer?
Yeah.
For example, if the kids inherit their family's genotypes, then if certain genotypes have implications for some kind of phenotypes, then those will be confounded with the environments that those parents provide to their kids.
Yeah. So I think the example that you're raising is that there may be some genotype that affects the kind of parenting environment. Actually, why don't you say a little more, say more what you had in mind. I'm not sure if I got it, actually.
So say that the kids' genotypes, so the kids' genetic information are partly inherited from their
Yeah.
parents, and certain genotypes have implications of phenotypes. Then if their parents have that genotypes and their kids also have that genotypes, then even though we control for ethnicity, then the effect inherited from those kids' parents are still not controlled for.
So I think the kind of example you have in mind is that there's maybe some genotype that is associated with the way that the kind of nurturing environment that parents create for their kids, and good environments that you create within a family lead to better educational outcomes among the kids, and those genotypes get transmitted to the kids. And so so then the kids have an association between those genotypes and their educational attainment, but those are actually genes that matter for nurturing environment, not genes that directly matter for educational attainment. Okay, so good. That's a great example. Yeah.
I have a question about, so you said that conditional on parents, like genotypes are literally randomly assigned, and like I understand what you mean is that there's no way that in one child it's correlated with sort of their neighborhood environment or something like that. But I don't know very much about biology, but to what extent should I think about, it's not a computer algorithm shuffling these different alleles, right? There's differential linkage disequilibrium at different points.
Yep.
So how should we think about is it literally randomly assigned, or is there just sort of a weird subset of genotypes that are more or less likely conditional on parents? Does that make sense?
Yeah.
Can I answer?
Yeah.
I don't think the issue is that there's chunks of the genome being shuffled because they're still being shuffled randomly. I think one could imagine a violation of this because we only observe the children that are born. So you could imagine a stressful prenatal environment that's acting as a selection gradient. So let's say a mother is, 20 when she has her first child and has a better uterine environment, so it allows a more frail kid to come to term and be observed by us. And she has her second kid at age 40, and it's a more harsh uterine environment. Egg quality has deteriorated, and that's a more stringent selection on certain genotype of the kid. So you could imagine there's be some correlation with some within family factors or stressful time because of poverty or whatever, but I've never seen it. I mean, it'd be interesting. You'd have to have enormous, like all of Sweden and Norway and Denmark put together all genotyped in the registries to try to uncover that kind of effect, because we think it's really, really small. But we know that, for example, sex ratios are affected by maternal age and stress. So it would be plausible that what we observe is not completely random, even within families in terms of genotype as well. But again, there's no existing evidence, and probably a very trivial effect in what we're kind of considering. But I don't think that the fact that genes travel together in linkage is a problem for the assumption of randomness.
Yeah, I mean, you can ask. Let's come back to this question when we have Fleur's lectures on the biology, and we can quiz Fleur on how random it really is, and where there might be violations of randomness. But I think that for everything we're going to be interested in, it's basically random. Okay, so this is actually, I mean, it's a good exercise to think about other examples where even within ethnicities you can't necessarily infer causation. There could be examples that are similar to the chopsticks example, just within ethnicity examples, where subgroups within an ethnicity have different cultural practices that are correlated with different gene frequencies. And so it doesn't have to be as extreme as chopsticks use, but that kind of example also carries over. Okay, so now we're going to move on, and I'm going to derive what we mean by additive effects and non-additive effects of genes. And what's different from what you did in problem set 1 is you did it in problem set 1 for a single locus model. What I'm going to do is just show you what that same derivation looks like when we do it genome-wide rather than at a single locus. Yeah?
So for the control variables that you include, are there control variables that you should not include, specifically that have kind of flagged through research?
Yeah, for example, we would not want to include any control variable that
mediated effect, isn't it?
yeah. So anything where the effect of the genotype might work through a channel, you wouldn't want to control for that, because then you're eliminating part of the effect of the genotype. For example, let's say that a gene, particular genotype, leads you to be more, have a certain personality, more openness to experience, say, and that helps lead to more education. You wouldn't want to control for that personality trait, because then you're only getting the part of the effect of the gene that doesn't operate through openness to experience. So
But if that's the question you're interested in, then maybe, that's
mediation.
Like,
Yeah.
You should understand that the interpretation of the coefficient is going to depend on what you control for.
Yeah, thanks. Good. So in what I'm going to talk about next, we want to be talking always about these causal effects. And so we only can talk about them when we've correctly conditioned on Z. So in everything we're going to talk about, you should imagine we're conditioning on Z, but for simplicity, I'm not going to show it. It'll make the notation more complicated to have that in there. Yeah.
Along the lines of what Patrick said, suppose I wanted to isolate the effect of parental genes on somebody's outcome and take out the effect of the person's own genes on the outcome. How would you do that? So I want only the effect of the parent's genes, not the effect of your own personal genes.
So if you have the children's and the parents' genotypes, you can observe the alleles that weren't transmitted, and those are going to be your treatment variables.
Yeah. So there's some recent work along these
Yes,
lines.
there's some unpublished work by folks at Decode in Iceland that do exactly this. The reason it hasn't been, it's a great idea, it's a brilliant idea actually, and the reason it hasn't been done much is just that it requires very large samples. But we're getting there. It's actually one of many, it's a very nice illustration of the kinds of things that are becoming possible right now.
And what they're finding, I think, in their preliminary results is that it seems to matter.
Yes. So the specific result that Augie has is that the maternal non-transmitted alleles sort of appear to affect children's outcomes independent, even if they were not transmitted, and probably through some environmental channel.
Yeah, so exactly the sort of example that Hui gave, we're now finding evidence for. Okay. All right. So we're going to start with this just general setup. We just have a population where we have, for a bunch of individuals, a measure of the outcome, and we have a bunch of these, and we have for every individual the set of genotype vectors. And we're just going to find for every genotype vector in the population, we're going to find the average level of the phenotype. That's the conditional expectation. And we're just going to normalize by subtracting off the average level of the phenotype in the population. So these are deviations from the average. And this function of the genotype, which is giving us just, given your genotype, how on average does your phenotype differ from the average in the population, that's what we call the genetic factor. This function of the genotype, G, that's the genetic factor. Anything else is something that we can't predict based on genotype. It's something where your genotype wasn't informative about that variation in the outcome. Okay, so that's the part of the outcome that's not genetic. Now we can always write our outcome variable we rewrite it in terms of deviations from the mean, so we don't have to carry around constants. And then this demeaned version of our phenotype is just going to be this genetic factor, the part that we can predict based on your genotype, plus the part that we can't predict based on your genotype, the residual. And by construction, this residual is going to have mean 0, conditional on your genotype, because anything that you could have predicted based on your genotype is part of G. So we can't, there's nothing we can predict that's not in G. Okay, now what we're gonna want to do now is take this genetic factor and decompose it into a linear additive piece, which we'll call the additive component, and then the stuff that's not additive, the non-additive component. So how do we do that? Well, we're going to define the best linear predictor of your phenotype based on your genotype vector as the set of coefficients beta that give you the best fit to your outcome. So this is just the population regression of the outcome on the genotype vector. And the best linear predictor is this population regression function. So it's a linear function of the genotype vector. And that's the linear function that minimizes the squared deviation, the sum of the squared deviations from the truth. We know from regression theory what this beta is going to be. It's x prime x inverse x prime y. Okay, so that's our best linear predictor of the outcome based on the genotypes. And so this genetic factor, which as we defined above, is this what you can predict the conditional expectation of the outcome given the genotype. We're going to now write this. We're going to add and subtract the best linear predictor. So now this term let's start with this term on the right. The best linear predictor, d-meaned, that's what we're going to call the additive factor. And the term on the left, the conditional expectation minus the best linear predictor. So the deviations of what we can predict based on your genotype that's not captured by the best linear predictor, that's what we call the non-additive genetic factor, n. Okay? Now these two things are mutually orthogonal to each other by construction. One way to think about that is that this additive factor is this best fitting linear regression function. So what's not captured by it is a residual. And we know by construction, by regression theory, that anything which is, that the residual is going to be orthogonal to the best linear predictor. So we had before that the d-mean phenotype was the genetic factor plus the residual. And now we have the genetic factors, the additive component plus the non-additive component. So now we can write the population model this way. That is the additive component plus the non-additive component plus the residual, where the residual has mean 0. And all three of these are mutually orthogonal to each other. Okay, so that's the key equation to keep in mind. That's the key fundamental model of genetic effects, where these things are interpreted. These are the causal effects of the genotype because we've conditioned on Z. You could think of it as this is all within one big family. Maybe these are all siblings in this population. So all of their genotypes are random. And what we're seeing is the causal effect that can be captured by an additive piece, part of the causal effect that's not additive, and then a residual that we can't explain by genes. Now we can actually go further with this kind of decomposition. We can decompose the non-additive component. So you could decompose it into a dominance component, which is the nonlinear effects of a genotype at any particular variant. So that's that a regression function requires that going from 0 to 1 has the same effect as going from 1 to 2. And there's no reason why those need to be the same. We could allow them to be different. And the deviation from the best fitting linear effect is captured by the dominance term, and there's one of those potentially for each variant J. And then there would be a set of, so another restriction that the linear form imposes is that there's no interactions between different variants. But there could be interactions between different variants, and there's many different kinds of interactions there could be. There could be, these are called gene-gene interaction effects sometimes. There could be an interaction between the additive component at 2 variants, between the dominance component at 2 different variants, between the additive component at 1 variant and the dominant at another, between the additive components at 3 different variants, and so on. You could keep going. There's many, many, many potentially of these epistatic components. Okay. Now, what we're going to often do is, and we'll get into the reasons why, it has to do with estimation and the fact that multiple hypothesis testing is going to be a big problem, and there's many genetic variants to look at. It's often going to simplify our lives to focus on just the additive model. So we want to understand what that means when we focus on the additive model. Are we assuming away all of this other stuff? Well, not really. Here's what we're actually doing. We're saying that we're writing this equation from before, the additive plus the non-additive plus the residual, and we're grouping together the non-additive and the residual and calling that a new residual, epsilon. So why can we do that? Well, we know that this non-additive component has mean 0, the way we've constructed it. Oh, something's going crazy. I don't know. Okay. Where were we here? There's some funny hand on the screen. Okay. Is this? Yeah. Okay. So this epsilon has mean 0 because of the way we've constructed the non-additive component and the residual both have mean 0, and they're both orthogonal to the additive component. So their sum is orthogonal to the additive component. And so this is a perfectly legitimate model to study, and we're not assuming away anything. We're just calling that part of the residual.
I don't know what's going on.
Here. Okay. Or here's the full model when we don't suppress the covariates. So we have the Z's in the regression as well. So this is often the thing that we're going to be dealing with when we come to estimation. All right. OK, so once we have this additive model, what are these betas that we focus on? How do they relate to these causal effects that we're interested in? Well, we call this vector of betas the vector of genetic effects. And remember that, we can interpret variation in these x's as random. So these betas are the average causal effect from this hypothetical experiment. So beta j in particular is the average causal effect of modifying variant j at conception. So we go from 0 to 1 or 1 to 2. And beta is the average causal effect. And in what sense is it an average? Well, it's averaging over individuals in the population. It's averaging over the dominance effects and the epistatic effects and the distribution of genotypes, which combine with the epistatic effects to potentially change the effect of the gene. So it's an average over a lot of things, but it has a causal interpretation nonetheless. Okay? Yeah.
Is it really a causal interpretation? I mean, it seems like you can always argue that there are things that you omit, right? So in the example that you were giving, like, let's say, if you're controlling for parents, but the children have different ages, then, of course, different ages already may imply that you have different family history in a sense. So time-varying family characteristics may be affected.
Yeah.
Even if the children are twins, then exposure to different set of so it seems like in the even though the epsilon is orthogonal, you may still have omitted.
Yes. It's a good point. I want to be clear. What I'm talking about here is the population model. So I want to imagine that the set of z's. So in the example, I want to imagine that the set of z's includes everything that we need to control for, such that x is as good as randomly assigned. Okay. So if there's an issue of the kids being different ages and that matters, then I want the z's to include the year in which the person was born. It's a good point. So we should be clear that the z, just conditioning on family doesn't necessarily solve everything. There may be other things that need to be included in the z's. But but once we've done that, then we can interpret the betas as average causal effects. Now there's another question, which is when you come to an actual application where you're estimating, then have you controlled for everything you need to control for? And then we can, then we have to have exactly this conversation of, are age effects important and should they be in the model?
Is it clear that you need to control for the age effects there? Because the gene, what's happening with the gene is random in relation to whether the kid is older or younger. So leave aside some of these selections.
Only the issue that I raised before, if there's just a like trisomy 21, as an example, is correlated with maternal age. Only to the extent that genotype is correlated with these within family factors, which, again, for common variants, we have no evidence that common variants we're studying, we have no evidence so far that they are. Of course, I think it may be actually more dangerous to condition on, say, child's age because you introduce collider bias because there's differential behavior in the parents based on the quality of the first child. They may or may not have a second child. They may control their fertility. They may invest differently in different kids. You start to introduce within family behavioral consequences of the kids' genotypes. I mean, it's a complicated issue. I'll talk a little bit about it in the next talk.
Yeah, so I agree with that. So I think I was wrong in the answer to the question. The right answer is if you condition on parents, then the genotypes are randomly assigned, and these are average causal effects. It may be that the causal effect is different for someone who was born earlier than someone who was born later. And that's part of what's being averaged over in the average causal effect. And we might want to then, we might want to look at age-specific causal effects. That, it could be the research question you're interested in. But, I think they still have a causal interpretation even if you don't condition on age in that example. Okay, yeah.
Sorry, I guess what he had in mind is not maybe age per se, but there's an environment, you know, in which each kid was born. Because, for example, if mother was really young when she was giving birth to the first kid, and she was very stressed, and that might affect, you know.
But again, only till if it's predictive of the genotype. So if it's a spurious confounder of genotype, that's the only way that this introduces bias. So you could make an argument for that, but you'd have to show it empirically.
Well, it changes the interpretation of the causal effect because it's now an average of causal effects of the genotype over different environments in which a child grew up. But it's still a causal effect. It's just a different average causal effect.
But then, if, like, for example, sorry, I don't know much about the biology, but if mother is more stressed, and then that causes certain genes to be more active compared to the others, and that might also change the interpretation of the causality, right? Because then there is a third factor that's driving both the genes and the educational attainment.
Well, right. So, well, as Dalton said, it only can make it not a causal effect if it causes the genotypes to not be randomly assigned anymore. If it causes a selection bias in which,
I think all of these examples that we're coming out with are things that are leading to a difference in the beta, and so there might be heterogeneous beta in the population where one person, the effect of the gene is different because of environmental stresses or things like that. But the type of biases that we're worried about here are things that change the expected value of X. And so if the environment is changing what we expect X to be, so for example, a person coming from a different ancestry, then maybe certain genotypes are more common, then that's going to break our causal interpretation. But if it's just things that lead to heterogeneity in the effect of the SNP, then we're just going to be averaging over all of those kinds of things. And so we still, again, I think that's, you think about things that affect beta and things that affect X. And things that affect beta are fine. Things that affect X are going to cause problems.
So this is great. This is exactly the kind of discussion that we want to be having now and also over the next 2 weeks, because I think some of these issues of interpretation are exactly the kinds of things that many people get confused about, even researchers in the field. And so we want to make sure that we all are on the same page and understand correctly what we mean by causal effects and what we need to control for and everything. So this is a fantastic start of that conversation. I want to now, given that we're at the point where we're supposed to stop and I have a little bit more I want to tell you, I'm going to just finish in the next 5 minutes and then we'll take a break. So I want to say we're going to talk about we're going to largely treat the additive model as our benchmark model, but we're going to deal with lots of various extensions of this model and relaxations throughout the course. We're going to talk about dealing with covariates, dominance effects, epistasis, gene-environment interaction, which is not in the additive model, gene-environment correlation, which could be in the model, but we'll talk explicitly about it, as well as various additional assumptions. OK, so let me just give you a brief preview of what the next 2 weeks are going to look like. So today is about payoffs for genetic data, why we should care in various social sciences. The point here, so I want to warn you, or just be clear, I understand that many of you don't yet have much background in genetics. So there may be things that come up today where you just, you don't yet have the background to fully understand it, and that's OK. So don't worry if you don't know what, polygenic scores are well enough to, understand the everything people are going to say, but you will. And the point here is to show you, what the, how this can matter, and to, get you excited about learning about these tools over the next 2 weeks. So then tonight we're going to talk about the ethical issues, the historical misuse of using genetics, mixing genetics and behavior, misinterpretation of results, and IRB and privacy concerns. Day 2 is going to be, actually I should also say later today, there's a discussion of problem set 1. So, so there's going to be discussions of all of the problem sets throughout the 2 weeks, and those are going to be sessions where we have a chance to talk about any remaining questions that you have about the problem set. So make sure that before the session you've looked through the answers and think about whether there's anything that you still don't fully understand or you think is worthy of further discussion. It's a great opportunity to have more of the conversation, for example, we just had about the interpretation of causal effects or other conceptual issues. And we'll all be here, all of the instructors will be here for those discussions. OK, so day 2 will be largely about behavior genetics, where the goal, what we're going to do is treat this additive genetic factor as a single unit and estimate heritability, which is how much does that additive genetic factor matter? What's the variance explained in the phenotype by that additive component? That's going to be useful in terms of learning more about this causal model, understanding it more deeply. It's also going to inform research we're going to do with genomic data, because it will give us an upper bound on how much predictive power we can get from the molecular genetic data. Starting on day 2 and extending into day 3, Fleur is going to be giving some lectures on molecular genetics. And there's various reasons why it's important to understand the basic biology. We need to understand the modeling assumptions we're making. For example, the assumption of random assignment. How reasonable is that? Where might it break down? What kind of new data might become available that's relevant for genetics research? Epigenetic data, for example, or gene expression data. How do we think about how that relates to the genetic data and genetic processes? We ultimately want to learn about biological mechanisms through which genes matter for psychological characteristics and then ultimately behavioral phenotypes. And we'll also learn about environmental interactions with genetic mechanisms at the molecular level, so gene expression, for example, and epigenetics. Day 4, which is Thursday, is going to be focused on gene discovery. So the question of how do we actually estimate these beta Js? That's important to evaluate the reliability of published results. And it's also we're going to talk about the challenges in this kind of work, which are pretty general challenges, the same challenges that apply in a lot of other areas of social science. So I think the lessons here are going to extend much more broadly beyond just gene discovery, as we talk about power, multiple hypothesis testing, and population stratification, and how we deal with those in the specific context of gene discovery. OK, so then days 5 and 6 are going to be turning attention from the individual genetic variants to the aggregate information contained in many genetic variants. And we're going to talk about constructing polygenic scores, like a predictor of this additive genetic component. We're going to talk about genetic correlation, which is a question about how do the genetic effects betas on one outcome relate to the genetic effects for another outcome. You can think about this vector of genetic effects beta. What's the correlation between that vector for 2 different phenotypes? Do genes that have a big effect on cognitive ability also tend to have a big effect on educational attainment or not? And we'll talk about partitioning, which is about whether the genetic effects tend to be larger in some categories of genes than others. Like we could partition by biological function and say genes in the central nervous system versus other kinds of genes, and how do the genetic effects compare. Starting on day 6 and extending into day 7, we'll also talk about environmental factors and how they interact with genetic effects. So either modifying the effects of individual variants or modifying the effects of the genetic factor overall. We'll also talk about Mendelian randomization, which is a set of tools for potentially trying to use genomic data to estimate causal effects of environmental factors. And then Tonu Esko will give a lecture on epigenetics, functional genomics, and life course development. Matt Keller is going to be visiting, and he'll give a lecture on genomic relatedness, which is about estimating the similarity between two different people based on their genomes. So taking the whole genomic vector for individual I and individual I prime, saying how similar are those vectors to each other. That could be interesting within families to look at like this, amount of similarity between, say, siblings, or between people who are unrelated to each other. Maybe people who are married to each other, or maybe just people who, are complete strangers. And that can then be useful. These various measures of similarity can be useful for getting other estimates of heritability using the molecular genetic data itself. Now, on Wednesday and Thursday next week, we're going to come back to applications. So we're going to go into detail on several applications of the tools that you've been learning for the 2 weeks. Some of the same kinds of things that are going to come up today, and see how those tools get used in practice. And we're also going to then have a chance to talk about some of your research ideas. And we're going to talk about what the future looks like for genomics research and social science genomics in particular. Okay, so some of the themes that are going to come up throughout the 2 weeks are that we're going to try to drill this point that it's only worth doing an empirical analysis if it's well powered, because if it's not well powered, you don't learn much, no matter how it turns out. We're going to focus on 3 central challenges that come up again and again across in gene discovery, but also in applications of power, multiple hypothesis testing, and population structure or stratification. And another important theme about the interpretation of genetic effects is that they often operate through environmental channels. So we have this random assignment of genotype that leads ultimately to some outcome, but we haven't said anything about the channel through which that works, and very often it will be through environmental channels, and that has important implications for the interpretation of results. Okay, so let me not say much, given that we're over time, on uses of genetic data in economics. You'll see a lot of examples of this throughout the 2 weeks. So I guess there's no need to take much time on it now. In fact, I think actually Dalton's going to touch on a lot of this when he talks about sociology, and David will talk on a lot of it in political science. So let's take a break now for, it's 10:50. Maybe let's take a 15-minute break and resume at 11:05.